Yes. Yes, sir. Start the class, sir.
Okay, sir. Thank you. So, good morning, all of you. So, today we are discussing about the respiration in plants. So, first, understand the respiration. What is the meaning of respiration? Generally, we know what is the uptake the oxygen and release the carbon dioxide. These are called as respiration. In a respiration, so release the energy, and these energy is comes from the some food materials, like they are called as respiratory substrates. So in a plants, generally the plants are contain the what is that, chlorophyll pigments. So let us, we are taking to the one uh, diagram also in this. So let us you are understand here. So here, what is that? This is the plant. So plants are observe the minerals and from the root hairs and the what is that? They in the presence of sunlight, so they can uh, observe the energy from the sunrise and involved into the photosynthesis, and they prepare the food material. So in these, so food material means what is that? Generally uh, glucose. And glucose is, you are observing in the leaf, they produce the food material in the presence of sunlight. So these, that's why this is the, what is that? Photosynthesis. So the green plants are called as what is that? Autotrophs. So means they prepare the food material by their own. Then after you are observing, so these glucose is and the formed here, and then after taken to the oxygen and released to the carbon dioxide. So and similarly during these process and also some released the energy. So some energy is released in the form of heat. So that's why in a respiration is a specific continuous biological reactions in all living organisms. So it is not a only gases exchanges process, but involves our oxidation and reduction. So that is important. So next, in this, so now we discuss the cellular respiration. So what is the cellular respiration? Means, so the respiratory substrates can broken. So in these require the some energies. So means what is that? In involve the biological oxidations of organic molecules, breaking of the in a what is the respiratory substrates, breaking the carbon compounds and results in the release of the some energy in the form of ATP. So it occur in the cellule. So that's why it is called as cellular respiration. So next, in here, ATP is found in all living cells. It is called as universal energy, universal energy carrier or energy currency. Means what is that? The energy is formed in the form of ATP, adenosine triphosphate. So that's why it is called as energy currency. So ATP is formed in the mitochondria, formed in a cellular respiration organelle. So that's why in a previous classes we know one word, mitochondria is well known, known as the powerhouse of the cell. So in these, so let us we understood that this mitochondria diagram. Here in a mitochondria diagram, you are observing it is a complex structure. So it contains the two membranes, outer membrane and the very smooth, and inner membranes shows the a number of vastigations or folds. So these folds are here, these folds are called as matrix. So inner membrane between the spaces is matrix. In these folds are called as what is that? Cristae. So these folds are here, the cristae, and 70s ribosomes are also there. So in the cytoplasm, so in a mitochondrium, cytoplasm is in a cytoplasm glycolysis process is occur, and in a matrix generally in a matrix Krebs cycle is occur, and similarly cristae, the folds are there in a cristae. In a cristae, the electron transports are occur. So in a cellular respiration means the involve the biological oxidation of organic molecules, means require the energy for the breakdown of the substances of the carbon bonds and result the in the release the some energy in the form of ATP. So let us respiratory substrates. Oh, what is the respiratory substrates means? So which are involved into the oxidase during the respiration. So they are called as respiratory substrates. So means the compounds that are oxidized during the respiration are called as respiratory substrates. So respiratory substrates is main source. General common respiratory substrate is generally what is that carbohydrates. So based on to the respiratory substrates, it is classified into two types. One is the floating respiration. So that is means when respiratory substrate is fat or carbohydrates. In this next one, protoplasmic respiration. So protoplasmic respiration means when respiratory substrate is protein. So in the first one is what is that floating respiration. So the respiratory substrate is fat or carbohydrates. And protoplasmic respiration is what is that respiratory substrate is protein. So here we take the some examples, carbohydrates, banana, pulses. So these are the, what is that, some are substratory respiratory substrates. Respiratory, simply you understand what is the respiratory substrates means, which are oxidized during the respiration for the releasing the energy. So that substrates are called as respiratory substrates. So generally the respiratory substrates are different kinds are there. They are the generally carbohydrates and proteins and fatty acids, organic substances. These all also based on to these, so classified into two types. They are the what is that floating respiration and the protoplasmic respiration. Floating respiration means it is important floating respiration. So the substrates are fats or carbohydrates. In a protoplasmic respiration is substrates only what is that proteins. So that is the next one, just a definition, respiration. So respirations are classified into generally two types. So aerobic respiration and anaerobic respiration. So let us, we are taking to the respiration, aerobic respiration in the presence of oxygen. Here clearly you are observing C6H12O6. So 6O2 and gives rise to the 6CO2 plus 6H2O plus ATP. So in a glucose, here glucose combined with the, or these glucose is converted into what is that carbon dioxide and water and released the some energy. So the energy is probably 686 kilocalorie energy. So here energy is released here. This part is probably how much, 686 kilocalorie energy. In this reaction, the bonds between the carbon atoms will break down and energy will be liberated. How much? 686 kilocalorie. So next, in these, so it is a considered as an catabolic and exergonic reaction. Why? Because catabolic means simply you are observed the glucose. The glucose is the complex compound. These glucose compounds are converted into simple compounds like carbon dioxide and water. So the what is that? The complex compounds are converted into simple compounds. So that process are called as catabolism. So anabolism means photosynthesis is anabolic reaction, constructive reaction. Constructive reaction means these simple compounds are converted into complex compound. But here the complex compounds is converted into what is that? A simple compounds such as carbon dioxide and water and releases the some energy. So according to this equation, we understand the what is that simply saying respiration means. So which life process is releasing to the energy, that life process is called as respiration. So in this, the energy is released. So the energy is, what is that? Energy is released. Energy is released here. So that's why these are called as, what is that? Exergonic reaction. So next one, In these already know what is that the complex compounds. So these are the complex. Glucose is complex compound and converted into simple compounds. So it is the respiration. So next one in a aerobic respiration and anaerobic respiration. So in a aerobic respiration passes by the four steps. Mechanism is four steps. One is the glycolysis. Second one is the pyruvic oxidative dehydrogenation, and third one is Krebs cycle, and fourth one is the electron transport system. Let us now we discuss the glycolysis. So glycolysis means, so it is a very important for the 8 marks, and it can be involved into the aerobic respiration and the anaerobic both respiration. It can be occur glycolysis. The glycolysis word is has been derived from the Greek word that means glycose means the for sugar, and lysis means for splitting. So what is that? Sugar splitting. This means glycolysis. It is also known as EMP pathway. It is also known as EMP pathway because of a reason. The three scientists are, so that persons are there. So first person is a Gustav Emden scientist, and second scientist Otto Meyerhoff, and third scientist is J. Paranas. So these three scientists, famous scientists, are done the work on the glycolysis. So hence their name is given to the glycolysis pathway. So that is the scientist first name, first letter, like what is that? E. Emden, and the Meyerhoff, and the P. Paranas. So based on to these working recognition purpose, the glycolysis pathway is given name as what is that? EMP, EMP pathway. So these scientists are worked on the glycolysis. So in a glycolysis, generally these glycolysis occur in the cytoplasm of the cell. So that is a very important cytoplasm of the cell, important beta. Next one. In these, the glucose is undergoes partially oxidation and to form the two molecules of pyruvic cell. What is that? So the six carbons of one molecule of glucose is undergoes to and cleavaged into and formed into two molecules of pyruvic acid. So each pyruvic acid contains the three carbons. This process is called as glycolysis. Glycolysis means what is that? The glucose, the six carbons of one molecule glucose is converted into two molecules of pyruvic acid. Each molecule, each pyruvic acid contains the three carbons. So that process is called beta glycolysis. First one, what is the glycolysis? Is a question very important. Sometimes we asked not the glycolysis, we asked as what is that? EMP pathway. So EMP pathway means what is the Emden, Meyerhoff, Parana scientists are done the work on the glycolysis. Hence, the cycle name given as EMP pathway. So glycolysis means what is that? Important point is what is that? The glucose is undergoes to the and partially oxidation and to form the two molecules of pyruvic acid. So glucose is what is that? Six carbons, and pyruvic acid, each pyruvic acid is contain the three carbons. So let us come to the steps of glycolysis. So carefully observe beta. So in this first, so here carefully you are observing. First, in a first step, glucose, one molecule glucose here, the glucose is converted into glucose 6 phosphate. What is that? The glucose 6 carbons glucose is converted into glucose 6 phosphate. So this reaction is catalyzed by the hexokinase enzyme. Catalyzed by the what is that? Hexokinase enzyme. So in this reaction, the ATP is converted into ADP. Means these from the ATP, adenosine triphosphate, this phosphate is uh, added to the which one? Glucose. So that's time it is converted as what is that? Glucose 6 phosphate. So in this this phase is called as investment phase. Means what is that? ATP is utilized and added to the substrate. What is that? Glucose. So that's why it is called as investment phase. So these process is called as what is that? Phosphorylation. These phosphate. So the glucose is taken the phosphate from the ATP. Means here ATP is a phosphate donor. So ATP is phosphate donor to the glucose. Then hence here formed the what is that? Glucose 6 phosphate. So these process is called as what is that? Phosphorylation. So it is catalyzed by the enzyme is hexokinase beta. What is that? Hexokinase. So next step you are observing the glucose 6 phosphate is there. So from the glucose to form the what is that? Beta glucose 6 phosphate. Here the glucose 6 phosphate is again isomerized and as a what is that? Fructose 6 phosphate. So here also 6 carbon, here also 6 carbons. So carbons are similar, but only physical structure is changed. So glucose 6 phosphate and fructose 6 phosphate. So this is the isomerization. What is that? Isomerization. So in this isomer, what is that? The glucose 6 phosphate converted into isomer fructose 6 phosphate beta. So here also 6 carbon, here also 6 carbon. Here this reaction is catalyzed by phosphohexose isomerase. What is that? Phosphohexose isomerase. So next the isomer fructose 6 phosphate similarly converted into the fructose 1 comma 6 diphosphate or bisphosphate. So in this reaction similarly, so the fructose 6 phosphate is obtained the phosphate from the ATP. It means here the ATP is donor of the phosphates. The ATP comes from the fructose 6 phosphate ATP and added to the phosphate, and then it is converted as what is that? Fructose 1 comma 6 biphosphate or diphosphate. Here also the ATP is consumed and the converted into what is that? ADP. Means here one. So two ATPs are consumed here, and these in these two reactions. So it is the phosphorylation. Here also phosphorylation. In these two phosphorylations process, the ATP act as what is that? Phosphate donor to the substrate. So here formed the what is beta? Fructose 1 comma 6 diphosphate or biphosphate, bisphosphate. Next one here clearly observe that the fructose 1 comma 6 bisphosphate is there. So how many carbons? All are here 6, here 6, here 6, here 6. All compounds are contain the 6 carbons, only one molecules. So here beta, then here carefully from here. So here fructose 1 comma 6 bisphosphate in the presence of aldolase, the cleavage as a what is that? So dihydroxyacetone phosphate, dihydroxyacetone phosphate, 3 phosphate. So and next one here glyceraldehyde 3 phosphate. What is that? Glyceraldehyde 3 phosphate. Here fructose 1 comma 6 bisphosphate. What is that? 6 phosphate beta. In the presence of aldolase, it undergoes the cleavage reaction. So cleavage means it is the cleaved as a two compounds. One is the DHAP. It is also dihydroxyacetone phosphate is also known as DHAP. And another one is the glyceraldehyde 3 G3P. So it is 3 carbons and it is also 3 carbon. Here beta you are observing. So next DHAP isomerization involved into the isomerization, and these 3 carbon compounds is converted into glyceraldehyde 3 phosphate. Means here clearly observe that already here glyceraldehyde 3 phosphate is formed, and then the dihydroxyacetone 3 phosphate also converted into glyceraldehyde 3 phosphate in the what is that? Isomerization reaction in the presence of the phosphotriose isomerase enzyme. So let us here formed the how many glyceraldehyde 3 phosphate are formed? Two molecules of glyceraldehyde 3 phosphate is formed. So already one is there, and here DHAP is converted or isomerized into the glyceraldehyde 3-phosphate. That's why here formed the two molecules, of the glyceraldehyde 3-phosphate. So after these process beta, here what is that? It is the clearly you are observing better. It is the isomerase reaction. So means the glyceraldehyde 3-phosphate is isomerized into dihydroxyacetone phosphate 3-phosphate. Similarly, the hydroxyacetone 3-phosphate is isomerizes into the what is the glyceraldehyde 3-phosphate. So these involvement, the catalyzed by the enzyme name is what is that? Phosphotriose. So here beta you are observing. First one, what is that? In this step, glucose. So what is that? Glucose 6-phosphate. So in this you should write the what in the catalyzed by the which one? Exokinase. This reaction is called as what is that? Phosphorylation. Next one here also what is that? Isomerization. And next in this reaction what is that? ATP is act as a donor. That's why it is also phosphorylation. So in a two steps are investment process. One is the glucose to glucose 6-phosphate, and another one is the fructose 6-phosphate to fructose 1 comma 6 bisphosphate. These means in these two reactions the ATP is act as a phosphate to donor to the substrate. So that's why these two reactions is called as what is the investment phases. So here fructose 1 comma 6 bisphosphate is cleavaged in the presence of or catalyzed by the aldolase enzyme and formed the dihydroxyacetone phosphate 3-phosphate and glyceraldehyde 3-phosphate. In this reaction, isomerization. In the isomerization, DHAP means dihydroxyacetone 3-phosphate is converted into isomerase as what is that? Glyceraldehyde 3-phosphate. Okay, now here formed the two molecules of glyceraldehyde 3-phosphate. So let us, you are observing in this reaction. So here the phosphate, inorganic phosphate, the source is inorganic phosphate is comes to the glyceraldehyde 3-phosphate and it converted and resulted to 1 comma 3 bis or diphosphoglyceric acid. So in the phosphate, what is source? It is not a inorganic phosphate came from the inorganic phosphate, but not a source is not there ATP. So and here NAD reduction undergoes to reduction process and gives to the what is that? NADH2 or write as a NADH plus H plus NADH plus H plus. So in the presence of what is that? Triose phosphate dehydrogenase enzyme. Triose phosphate dehydrogenase enzyme. It is converted into as a what is that? 1 comma 3 bisphosphoglycerate. So how many molecules are? Two molecules actually. So here you are observing 1 comma 3 bisphosphoglyceric acid is glycerate is. Then here also 1 comma 3 bisphosphoglycerate and converted into what is that? 3 phosphoglycerate. 3 phosphoglycerate. So these reaction so catalyzed by the a specific enzyme. What is that? Phosphoglyceric acid kinase. What is that? Phosphoglyceric acid kinase. So in these, so the ADP so is converted to ATP means involved dephosphorylation. These are called as substrate level phosphorylation. This reaction is called as substrate level phosphorylation. So in this converted into the ATP, in this process ATP formed. So where the ATP is formed, that process is called as substrate phosphorylation and comes from 1 comma 3 bisphosphoglycerate to 1 phosphate is added to the ADP and converted into the what is that? ATP. Here ATP is formed means this ADP 1 phosphate is added to the ADP. So these are called as phosphorylation. So dephosphorylation means what is that? From the substratum, the phosphate is added to the ADP and converted as diphosphate, converted into the triphosphate. So this is the dephosphorylation or substrate level phosphorylation. So next one here, the 3 phosphoglycerate converted into and two carbons of the phosphoglycerate. It is also isomerized enzyme in the catalyzed by the isomerized enzyme. So here, what is that phosphoglyceromutase? In the presence of phosphoglyceromutase. So then these two phosphoglycerate is involved into the dehydration. So means the water is released from the substratum. So water is removal. It reaction is catalyzed by the enolase enzyme. What is that? Enolase enzyme and gives rise to the phosphoenol pyruvate. What is that? Phosphoenol pyruvate. So PEP. Phosphoenol pyruvate is next undergoes to the dephosphorylation process. From in this, the phosphate is added to the ADP and converted into the or formed to the ATP. So means ADP plus phosphate is formed the what is that ATP. So what is that substrate level phosphorylation? Here ATP is formed. Then after it formed the pyruvate. The pyruvate is 3 carbon. How many pyruvates are formed here? Two. Any one, two, two, two. So here we are observing what is that in the two process. Here it is the one glucose to glucose 6 phosphate is one is the phosphorylation process, and next one fructose 6 phosphate to fructose 1 comma 6 bisphosphate process. It is also investment means here ATP is consumed, so ATP is lost here. But in these you are observing 1 comma 3 bisphosphoglycerate and the two phosphoenol pyruvate to pyruvic acid. So these two reactions are substrate level phosphorylation. Here the ATPs are formed. So let us, so simply in an examination you should learn the very clear only chart. Then after you should write the equation type. So that is the very important. Let us we discuss about the uh, what is that, how to write the reactions. So here you are observing. So in this first one you are observing first process is what is that beta phosphorylation. So phosphorylation we know what is that, the glucose and combined with the ATP and the in the presence of what is that catalyzed by the hexokinase. So formed the what is that glucose 6 phosphate and ATP is so consumed to and phosphate is donor, ATP is donor to the glucose. So that's time this glucose is formed as a what is that glucose 6 phosphate and ATP is one phosphate is goes to what is that glucose. So here formed the what is that ADP. So ADP. And next one isomerization. So in isomerization, so remember beta, the glucose 6 phosphate. These glucose 6 phosphate is so and isomerization in the presence of phosphohexose isomerase enzyme and gives rise to the what is that fructose 6 phosphate. So here 6C, here 6C. And next one phosphorylation. So in this also phosphorylation process, what is that here? The fructose 6 phosphate, here you are observing, the fructose 6 phosphate combined with the ATP. Means what is that? The ATP is act as a phosphate donor to the fructose 6 phosphate. That's time one phosphate is added to the fructose 6 phosphate. That's time it can be resulted as what is that fructose 1 comma 6 bisphosphate and phosphate is ATP is a phosphate donor. So that's why here formed the what is that ADP. So clear beta. So these one, the glucose, first one also phosphorylation. Here also ATP act as a donor. Here also ATP act as a phosphate donor. So these two are the what is that investment reactions, investment phases. So these two are. One is the phosphorylation and here first one phosphorylation, third reaction phosphorylation beta. Next, you are observing cleavage. Already we know cleavage. Cleavage, what is better? So fructose 1 comma 6 bisphosphate. So in the presence of aldolase enzyme, it is cleavaged as dihydroxyacetone phosphate 3C, and another one is the 3-phosphoglyceraldehyde. So DHAP and 3GP. These are called as 3G3. Next one, what is that beta? Isomerization. Isomerization means what is that beta here? So dihydroxyacetone phosphate is DHAP is converted into what is that glyceraldehyde 3 phosphate in the presence of what is that triose phosphate isomerase. So here already one glyceraldehyde 3-phosphate is also there. So you are observing. So 3-phosphoglyceraldehyde is there. So next one, what is the dihydroxyacetone phosphate also converted into glyceraldehyde 3-phosphate. So let us here form the two molecules of glyceraldehyde 3-phosphate is formed. So next one, oxidation. So oxidation in this your glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate is, so what is that, NAD plus is converted into what is the NADH. So here 1 comma 3 bis phosphoglyceric acid. So in the presence of glyceraldehyde 3-phosphate dehydrogenase. So that is the important. Next one, your glycer. What is that? Glyceraldehyde 3-phosphate to 2 gives rise. One is 1 comma 3 bis phosphoglyceric acid. Here NAD plus and gives rise to the NADH. Next reaction is so dephosphorylation. So in this dephosphorylation, here beta you are observing. So what is that? 1 comma 3 bis phosphoglyceric acid and ADP. So in the presence of which enzyme? Phosphoglycerokinase, and result the 3-phosphoglyceric acid and formed the ATP. Means 1 comma the phosphate is comes from what is that, 1 comma 3 bis phosphoglyceric acid, and these one is added to the ADP, and here formed the what is that, ATP. So this process is called as dephosphorylation. So this is also known as substrate level dephosphorylation. And next one, interconversion. So in interconversion, eighth reaction is just you are observing. So the 3-phosphoglyceric acid in the presence of are catalyzed by the phosphoglyceromutase. So these molecules is shifted to 3 carbons to here what is the 2 carbons. So these are the called as what is that, interconversion. So here resulted, so what is that, 3-phosphoglyceric acid is resulted in the presence of or catalyzed the phosphoglyceromutase. Here 2-phosphoglyceric acid is formed. And next dehydration. So dehydration also you know what is that, 2-phosphoglyceric acid is so lost the water in the presence of what is that, catalyzed by the enolase. Here 2-phosphoenol acid form is there. So next last one is the dephosphorylation beta. So dephosphorylation, what is that? Phosphoenol pyruvic acid here, phosphoenol pyruvic acid, PEP generally. So here also phosphoenol pyruvic acid, one phosphate is added to the ADP, and here formed the ATP. So this phosphoenol pyruvic acid is converted into pyruvic acid in the presence of catalyzed by the specific enzyme is what is that, pyruvic kinase enzyme. So in these reactions you are observing completely. So first you are observing. So here the first the glucose is similar you are observing. These one glucose is converted to, so last one what is that, converted to what is that, pyruvic acid. How many pyruvic acids? So clearly you are observing here. So here the glucose is converted to what is the pyruvic acid. This process is called as glycolysis. So in this reaction beta, now we write the simple. So here reactions, first you should write what is the glucose plus ATP and gives rise what is the glucose 6-phosphate plus ADP. So in the presence of exokinase, on arrow mark you should write the exokinase. Reaction is phosphorylation, and glucose 6-phosphate and gives to the fructose 6-phosphate, and fructose 6-phosphate plus ATP and gives to ADP and fructose 1 comma 6 bisphosphate, and fructose 1 comma 6 bisphosphate uh, in the presence of aldolase enzyme gives to the what is the dihydroxyacetone phosphate, and the here DHEAP and the here glyceraldehyde phosphate. Next reaction you should write the what is that, glyceraldehyde 3-phosphate plus NAD gives rise 1 comma 3 here diphosphoglyceric acid plus gives rise to the what is that, NADH2. And next reaction, how you write here, 1 comma 3 bis phosphoglycerate plus ADP and gives rise to the 3 phosphoglycerate plus ATP. In the present arrow mark, on an arrow mark, you should write the MJN, phosphoglyceric acid kinase. Next, you should write what is that 3 phosphoglycerate gives rise to the 2 phosphoglycerate. And arrow mark, you should write the what is that phosphoglyceromutase. Next one, 2 phosphoglycerate gives to the what is that phosphoenol pyruvate plus H2O. Plus means released H2O. An arrow mark, you should write the what is that enolase. Next, here uh, phosphoenol pyruvate plus ADP and gives rise to the pyruvic acid and ATP. So this is the way of write the reactions: phosphorylation, glucose, and isomerization, and the 3 phosphoglyceric acid, cleavage, isomerization, oxidation, so dephosphorylation, so in intraconversion. Next, dehydration. So 10th one, what is the dephosphorylation? So in this energy sheet, so energy sheet of glycolysis beta, first one, ATP produced by the 2 substrate level phosphorylation. So how many molecules are formed? 2 into 2, 2 molecules. So here formed the 4 ATP. And from NADH generated, so how many ATPs are formed? 4 ATP. So total 8 ATPs are formed from NADH to 1 NADH gives to the 2 ATP. So here 2 molecules of NADH so involved into the dehydrogenation. So that's why here formed the 4 ATP. So substrate level phosphorylation 2 are there. So that's why here 4 ATP. Total 8 ATPs are formed. And next, consumed ATP. So first process is the consumed ATP is the 2 ATP. So that's why net ATP is, you are observing, net ATP is 8 ATPs minus 2 ATP. 2 are consumed, 8 ATPs are formed. So 8 ATP means what is that? From the substrate to substrate level phosphorylation get the 4 ATP. So in a each substrate level phosphorylation formed the 2 ATP. So 2 reactions are there. So that's why 2 into 2. So 4 ATP. So NADH 2 into 2. Each NADH is here 4 ATP. So total 8 ATPs are formed. So net gain, what is that? 8 ATP minus 2 ATP gives to the total net ATP in 6 ATP. So here you are observing in a chart. So clearly you understand it in a, here better. So here ATP consumed, here ATP consumed. So ATP converted into ADP, ATP converted into ADP. So in these two reactions, ATPs are consumed. And here NAD dehydrogenated and formed the NADH plus H plus. Here these are from these reaction, all are 2 molecules. Remember it, all are 2 molecules from. So these two. So 2 molecules means one NADH2 gives to the 2 ATP. So here 2 molecules. So that's why 4 ATP. And here ATP. So but how many molecules? 2. So here 2. Here 2. So these 2, 4, and these 4. So total how many? 4, 4, 5, 6, and 7, 8. Total 8 ATPs are formed and consumed 1 ATP, 1 ATP. 8 minus 2 beta. So total 6 ATPs are net gain in the glycolysis. So that is the very important. So in this we are understand the glycolysis means what is that? It occur in the cytoplasm. So next process is what is that? No oxygen is used here. So in this, the what is that? No oxygen is used in this, and this is the common step for the both aerobic and anaerobic respiration. So these glycolysis also known as hexose diphosphate pathway. It is also known as what is that? Hexose diphosphate pathway. So it is a very important glycolysis for essay question type. So remember it. So hexose diphosphate pathway. So in a simply we understood what is that completely. The 6 carbons of glucose is converted into what is that? Converted into 3 carbons of 2 molecules pyruvic acid. So it is the glycolysis. In a 4 steps, so you understand that in a 4 steps, first one is the glycolysis, and second one is the oxidative decarboxylation of pyruvic acid, and third one is the Krebs cycle, and fourth one is the electron transport system. Okay. Here, oxidative decarboxylation of pyruvic acid. So second one. So in these, so in a end of the glycolysis, we get the what is that pyruvate or pyruvic acid. These pyruvic acid is undergoes to dehydrogenation and decarboxylation. So here you are observing NAD plus is converted into NADH, and one carbon dioxide also removed. So actually pyruvate is 3 carbon. So here one carbon is removed. So that's time 2 carbons of acetyl coenzyme is formed. Acetyl coenzyme is formed. So it is the oxidation reaction. So coenzyme A formed the what is the here 3 carbon of pyruvic acid is converted into 2 carbons of acetyl coenzyme A. So here you are observing so as a equation form. So pyruvic acid and combined with the coenzyme A and involved NAD plus and resulted what is that acetyl coenzyme A and resulted carbon dioxide and formed the what is that NADH plus H plus. So here this is the 3 carbon and it is 3 carbons and acetyl coenzyme A is the what is that 2 carbons. These acetyl coenzyme is interlinked between the what is that glycolysis and Krebs. So this is the pyruvic oxidative decarboxylation of. So in these, so you are
Thank you for
observing.
the session, sir.
Okay, sir. Okay. So 2 NADH is formed. So each worth is 3 ATP. So total 6 ATP is net gain. Okay. Okay, sir. Thank you. Thank you, sir.
Thank you, sir.
Thank you, sir.